Bacterial wilt is a destructive disease of cucurbits that can result in severe yield loss or at times complete crop failure. Susceptibility to bacterial wilt differs across cucurbits. Cucumber and cantaloupe are considered highly susceptible, while squash and pumpkins are less susceptible and watermelon is resistant. Early symptoms of bacterial wilt include individual leaves turning dull green and wilting. As disease progresses, entire runners or whole plants will wilt. At first, plants may partially recover at night, but eventually the wilt will become permanent. Foliage and vines will collapse, turn brown, shrivel, and die. Wilt symptoms may be noticeable in as few as 4 days from infection on highly susceptible hosts, but can take up to several weeks to become evident on crops that are less susceptible. Young plants often wilt and die soon after infection. A key diagnostic feature for bacterial wilt is the emission of a slimy, sticky ooze from cut stems. A field diagnosis can be confirmed using a simple bacterial ooze test. With a sharp knife, cut through a wilted but not yet dead vine on a section near the crown, touch the two ends together and then slowly pull them apart. Fine thread-like strands of bacterial ooze will be drawn out when the bacteria is present. This test works well for cucumber and muskmelon, but is less reliable for squash or pumpkin. Another option is to place cut pieces of symptomatic stems in a clear glass jar filled with water. If the bacterium is present, a cloudy string or mass of bacterial ooze will flow into the water from the cut stem pieces. Bacterial wilt is caused by the bacterium Erwinia tracheiphila. Striped and spotted cucumber beetles serve as vectors for this pathogen, carrying the bacterium from plant to plant during the growing season. The life cycles of the bacterial wilt pathogen and its vectors are closely associated, with the incidence of bacterial wilt being directly correlated to cucumber beetle population density. Cucumber beetles hibernate through winter under leaf litter and in other protected sites. The beetles become active once temperatures remain above 55 degrees in the spring. As soon as cucurbit seedlings begin to break through the ground, the beetles begin to feed on the cotyledons and later feed on the leaves, stems, and flowers. Striped cucumber beetle larvae also feed on root systems, causing damage that can result in wilt. Disease spread occurs when beetles feed on diseased plants and then feed on nearby healthy plants. The bacterium is deposited through the beetle mouthparts and in the frass deposited into wounds created during feeding. Once the bacterium invades xylem tissue, it spreads rapidly throughout the plant. The matrix of bacteria and ooze obstructs water movement in the xylem vessels, causing wilt symptoms. Close to harvest, a second generation of striped cucumber beetles may acquire the bacterium while feeding on infected plants still remaining in the field. Fall planted cucurbits may then be infected by this generation. These late season adults overwinter with the live bacterium in their gut and transmit the pathogen to young plants the following spring. Disease management of bacterial wilt heavily relies on management of its vector, the cucumber beetle. Disease management should begin with sound cultural disease management practices, including utilizing first resistant cultivars. Options are limited, but some moderately resistant cultivars are now available. Physical barriers and mulches can also help manage the cucumber beetle. Small plantings can be protected from cucumber beetle by using row covers, fine netting, or cones placed over small plants to exclude the beetles. Plastic mulches can also reduce overwintering beetle populations by reducing the larval survival. And reflective mulches have also been shown to repel the cucumber beetle at times. However, research is ongoing on the efficacy of these reflective mulches. It is also a good idea to practice crop rotation and rotate cucurbit crops on a 3-year cycle, or at the very least, avoid using highly susceptible cucurbits 2 out of the 3 years during this rotation to reduce the cucumber beetle populations. Weed management is also important as wild cucurbit species can serve as hosts for the vectors, the cucumber beetle, such as burr cucumber and wild gourd. To effectively manage bacterial wilt, chemical control is often required in addition to these cultural practices. Bactericides are not effective, and therefore chemical control focuses on using insecticides. An insecticide program should be started as soon as seedlings emerge or immediately after transplanting. This is critical for protecting small plants from beetle feeding and ultimately from bacterial wilt. There are two types of insecticides that can be used for beetle management. First being contact insecticides, which protect plants from beetle infestations by applications of foliar insecticides 1 to 2 times per week during peak beetle activity. Systemic insecticides can also be used and are applied as a single post-transplant soil drench and can provide up to 3 to 5 weeks of protection depending on the product. Trap cropping also pairs well with insecticide use, and commercial growers may have success in planting trap crops around their field perimeters. These are highly susceptible cucurbit cultivars, such as Blue Hubbard, which attract the cucumber beetle, and then insecticides can be applied to the trap crop and reduce the need for applications on the harvestable crop. For more information regarding bacterial wilt and its management, please contact your county agent.